Hello, everybody. This is Jacob Smoot, and the topic of my paper is going to be on domestication and selection, evolution of genetics in beta splendens and other species found worldwide. So to begin with my kind of overview of the beta splendens species, were first kind of documented way back in the 1300s. They're also known as the Siamese fighting fish. These fish are normally found in these highlighted areas that I have on this map that I've shown in Thailand, Cambodia, Laos, and Vietnam. They habitate warm water areas, as these places are tropical, and where there's a lot of dense overgrown plants that allow them to protect themselves. Like I said, they are known as the fighting fish. These are very aggressive fish towards each other, the males in particular. And so it's important to have that dense overgrown kind of plant area. This also is low oxygenated water, so the beta splendens species has evolved over time to create what's called a labyrinth organ, which is also seen in different fish species. And this is just an organ that allows them to go up to the top of the water and breathe atmospheric oxygen. This is important in breeding, as these fish are bubble nest breeders, which means the males will blow bubbles and make a giant nest full of bubbles where it will lure in the female and then be able to wrap around their body and take care of the eggs. So I'm going to kind of go over and start with the evolution of beta splendens. So it all started in 1920, the domestication of the beta fish through selection was first seen then. These pictures that I've included are examples of what this fish looks like. This is one of the most popular varieties of beta splendens. It's called the veil-tail variety. This was done collecting offspring that had longer fin types. And over time, they were able to perfect this longer fin, which created a veil look. And this was done in Thailand. To continue, for 40 years, the veil-tail was the hot fish on the market. You know, it was the most popular variety of fish that they were selling. But in 1960 in India, there were some breeders who were able to introduce genetic mutations, which caused this veil-tail to become a double tail, as shown in figure 2. There was a few complications, though, with this double tail variety, because the males had a shorter body and they were unable to successfully wrap around the female while breeding. And so they had to kind of play with the genetics a little bit more, and they were able to successfully breed with a veil tail and a double tail to make what is known as the delta tail. And that is seen on the right in figure 3. Delta tails are kind of like the next step up, but then further along in time, there was picture, like, let's just take a pause. The fin type on the delta tail is about at a 120-degree tail width. But over time, they wanted to successfully engineer a fish that could have a full 180 span, and that is what is the half moon variety. So on the left in figure 4, we have what's called the R39. R39 was created because after the creation of this half moon beta fish, there was many problems due to inbreeding, and so they were having issues with fitness within the species. So what they did, breeders worldwide came together and they started to introduce their half moon betas so that they could have more genetic diversity and not have as much inbreeding. The first offspring of this mating was what's called R39, and this is the template species for all half moon betas to today. So that's a big deal. That's like the Adam and Eve of the half moon beta fish. So it's pretty cool. You know, over time, this is all very recent. Like in 1975, this was happening. And so from 1980 up until today, there has been a huge increase in artificial selection and selection to make more and more species. I've included a few pictures of a few different types in figure 5 and 6. You know, you have your plaquette betas and you have your plaquette crown tail betas. I mean, there's a whole variety. There's a koi beta, there's a giant beta, there's so many different types of fins and body shapes that have emerged since R39, and they continue to form today. So we see that there is a large domestication that's happening to these fish. But the point of my paper is to really understand that this domestication process through selection, along with other processes, are causing problems within the species in the wild, within the aquarium, or in the domesticated forms. So my first topic that I have is on the fish hatchery. So I was able to go through and research a little bit about how fish hatcheries are having a effect on the beta splendid in particular. And it was very interesting to find that. Some figures is that every year about 3.5 to 3.6 million beta splendids are exported out to the world. This is a huge number of beta fish. So we can see that there is a commercial drive for this artificial selection to be taking place. There are many different fish hatcheries for these within Thailand, but this study was done in Nangkorn Pathum, Thailand, and it was very interesting. They used what's called a closed breeding system. So I'm going to go over that in just a minute about what that means. But the biggest purpose was they wanted to quantify the genetic diversity in beta splendids in the wild versus the genetic diversity within the fish hatcheries. They wanted to see if there was going to be any changes in the fish hatcheries and if those changes had any like negative effects. And so what we can say is with genetic diversity, there's going to be a higher fitness. So this is a figure that I was able to find within the research paper in figure 7 that explains the two different breeding systems that are used within these fish hatcheries worldwide. So there's the closed system, which I'll go over in just a minute, and there's the open system. The open system is just where partner fish hatcheries will come together and they'll bring in their brood stocks so that they can have a very large mixed amount of fish, which then they'll discard the females, they'll keep the males, and then they'll sell them. Now, the closed system is what was used in this experiment. The closed system does not allow for outside fish to come in until years in advance. So you have 25 males and 25 females. You keep those as your parents and you start breeding them and you create offspring. Now, with those offspring, you know, they take the females, they discard them, and then they'll take the males and they'll sell them. The reason why this is happening is because males are more colorful, they're more popular, and so they're going to make the most money. The females are a lot drabber. They do obviously make the eggs, but they're just not sold for as much, and so the commercial drive to have them is not as high. So I want to talk about these results that were in this study. Very interesting to find. So what they did is the scientists were able to take 23 females and 23 males that were wild beta splendens within the surrounding areas in Thailand, and they assayed them, and they were able to resolve 9 isozymes, which are just the enzymes that are found in the body that help with function. Um, some of them being like hemoglobin and just different enzymes that are really important for function. Um, so out of those um, 11 loci were be able to be resolved, and out of the five of those loci were found to be polymorphic. So that means that at those specific pieces of the genetic code, there was more than one allele. So there was like a big A and a little a. There was different alleles, or big A, big B. So what they found was that the wild population had a low heterozygosity. There was more homozygous individuals within the wild population. So then they took 25 male and 25 female fish from a local hatchery in Nacombe, Northampton, and there they harvested them, their liver and their muscle tissues, and they assayed these, and they were able to resolve 19 loci. So these areas in the genetic code. And they found that 7 of these 19 were found to be polymorphic. Again, saying that there's more than one allele at those locations. So the average heterozygosity in the wild species was found to be 6.5%, whereas in the domesticated forms had an average of 11.3%. Now this is huge. This is saying that for every 9 fish that you are selling, one of those fish is going to have a higher heterozygosity. Does that make sense? So one of those individuals will be heterozygous. This is very interesting because when comparing it to other studies that have been done on fish hatcheries that I was able to do, like on black sea bream, Japanese char, common carp, and guppies, which are all on the next slide, which I'll show you, they all had lower heterozygous rates versus the wild counterparts. And so this is just kind of establishing that within the beta splendens population, the homozygous individual is going to be more dominant. So that just establishes that actual low heterozygosity equals high fitness. So here in the figures, you can kind of see these are just the other fish that they were able to notice this in. So this isn't just the beta splendens that they're seeing this either this change in heterozygosity. So, and it's all due to the founder's effect, because the founder's effect kind of states that when you have a small population size, you're going to lose genetic diversity due to the low amount of alleles that are in the genetic pool. Another thing that I was able to kind of pull from this research paper was that there is possible error. You know, we can't always say this is a for sure thing, but there is a possible error in that they did have a very small research size. You know, they only studied 25 males and 25 females for this, but they did do this over a longer period of time. I think it was about 10 years. And so, I mean, it was a pretty substantial data. So I want to kind of transition now and go into different areas of where domestication and hybridization are causing problems within other species in our world, and kind of tie it back to the beta splendid. So first we're going to kind of talk about the Tibetan lotus plant and the honeybees. So within Tibet, the lotus plant flower is a very important flower for many reasons. One being that the honeybees there use this flower as one of their main food sources. So it's interesting to see that a lot of these apiaries are being formed, which in figure 12 shows kind of example. They're just like little bee homes. This is a way of domesticating the bees and having more control over them to be able to harvest their honey. So studies have been shown and have been done over the years, and it's very interesting to see that within areas of the apiaries, these Tibetan lotus flowers are decreasing their nectar volume by 60%. That's huge. That's a huge, huge amount of nectar that's being decreased. And this nectar is, like I said, the food source to most of the wild honeybees within Tibet. So there is a large decrease in the honeybee population in the wild, which is really bad. As we know, bees help pollinate, and if the plants can't pollinate, they can't create fruit or make more seed. And so there's kind of a negative effect on both ends. You know, the bees are being affected and the plant is being affected. And in general, as human beings, we are being affected by this because we need honeybees to be able to pollinate our crops. So this is a huge example, like the beta splendens, is how the domestication of an animal can cause severe consequences. So our next example is with the European wildcats, which have been shown to have a high hybridization with other domesticated cats in the areas. So this study was done in Sweden, Ireland, and these different studies showed that the wild cat population in these areas is becoming more and more endangered due to domestication of this species, and their genetic diversity is being lost completely. Now, this may not seem like a big deal, but it's just showing how us as humans can have a large, profound effect on a species and cause them to go extinct. And so for our third example, this one kind of goes back to the aquarium world, and there's a fish called the neon tetra. It's a very, very colorful fish. This fish is also a very popular aquarium fish, but it has also undergone extreme domestication and selection. So this is a schooling fish. I'm going to skip slides so you can kind of see in figure 15, this is what the neon tetra looks like. It's really pretty. This is a schooling fish. So you'll usually see these in schools of about 8 to 10 within the aquarium, but you can, in the wild, they're used to being in like hundreds just for protection and to keep optimal health. So what they've noticed is that the neon tetras in the fish hatcheries are showing higher mortality rates. So they're a lot more sensitive to aquarium parameters. So if you took like 100 fish from the wild and 100 fish from a captive bred fish hatchery, you're going to see a lower rate of death within the wild population. Now, a huge problem that is being caused right now is what's called the neon tetra disease. This disease, which is shown in this slide, in figure 17, it shows what happens. Now in figure 16 is the culprit to what figure 17 is showing. This is called Flavobacterium columnar, which is a bacteria that will enter in through the lungs and the mouth and will then just wreak havoc on the body. You'll see loss in coloration. You're gonna see almost like a scoliosis effect on the fish, like in figure B, and it will always lead to death. So it's very unfortunate because there's a lot of money being spent just to have fish die within a few months. The big problem with this bacterium is that it's mostly it's only within tropical species. So this includes the beta splendin. It's not only the neon tetra disease that it causes, but it can cause other problems within the beta fish. We're similar where they lose color, they're having that scoliosis effect. So the sad thing about this is that there is not a lot of research that has been done yet to kind of predict and to understand the pathology of this bacterium as much. You know, all they know is that the biggest thing is that they can prove is that there is a lower or a less, I should say, immune system within the domesticated fish. They're showing that they are not able to withstand these bacteriums that are normally found within the wild population. You know, if you go to Peru or to Argentina or to the Congo where you can find these different fish, you can scoop up the water and you're going to be able to find Flavobacterium columnar, but it's not affecting the fish like it is in the captive bred species. And so they can kind of conclude that they think it's a immunological disparity that's going on. And this is not only seen in the neon tetra, also the beta splendin, and in a lot of different other domesticated fish. So you can just see these slides again, just to kind of see the pictures. So, I mean, just to kind of conclude with my presentation, my biggest points I wanted to hit home was that domestication and selection is causing a huge evolutionary effect upon different species within our world. Our main species that we're seeing is the beta splendens, just because this one is the one that is kind of causing a lot of money to be put into it. All of these different species that I've talked about are effects of commercialization, people wanting money. And though there is a beauty within the species of beta splendens, there's also some negative aspects of it. It's very sad to see that a lot of these fish are succumbing to diseases a lot more easier just because their genetics aren't as sound as their wild counterparts. And what's also being seen is that, you know, the wild species population are being lost and their genetics are being lost due to catch and release programs and also the fish hatcheries releasing domesticated fish. So there's a lot of conservation efforts that are being done to control this, but they aren't as controlled right now, and there's not a lot of strict regulation on these. And so until these things get more controlled, we're not going to see much progression with the saving, I guess, of the genetics of the wild species, of the beta splendens in particular. And so I just want you to leave that kind of thought with you guys about the conservation efforts, and also hopefully that you guys can get a better idea of the effects of domestication on animals and their evolution through time. So this is just my reference cited page. And yeah, that's about it. But thanks for listening, and I hope you have a good day. If you have any questions, let me know.